Today, we are super happy to be launching an exciting new tool for the direct measurement of CO2 in the bioreactor. There will be two presenters today. I'm Kelsey Mado, a market segment manager at Hamilton for the biopharma market. I'll get us started with an introduction into the role of CO2 in the bioreactor and why it's considered a critical process parameter. Then Giovanni Campolongo will be discussing the tools that are available for measuring CO2. He is also a biopharma market segment manager for Hamilton Process Analytics. Throughout this presentation, please feel free to put questions in the Q&A. We'll answer some of them in real time, and then some of them we'll save for the end so you get a more thorough discussion. So we'll begin today with a brief discussion of how CO2 changes throughout a bioprocess and the importance of controlling it. We'll review the current technologies that are available for measuring CO2, finally leading to the development of a new dissolved CO2 sensor. Dissolved CO2 is always present in the bioreactor, but it can fluctuate significantly throughout a typical process. There are several factors that influence the level of CO2 at any given time. One of the biggest causes of CO2 increases in the bioreactor, at least in the case of bicarbonate buffer systems, which are typical to cell culture applications, is pH regulation. Whether you're adding bicarb to raise the pH or sparging CO2 to lower the pH, both of these things will lead to increases of CO2 in the bioreactor. Increases in viable cell density also increase CO2 in the bioreactor, both as a byproduct of cellular respiration and other metabolic processes, so protein production, for example. So CO2 will increase significantly throughout any typical bioprocess if it's not counteracted. The primary method for removing CO2 from the bioreactor is through deliberate CO2 stripping, which is done in order to counteract all of these increases. Now, in bicarbonate buffer systems, CO2 is closely intertwined with pH regulation, so you might imagine that tight pH control would naturally result in tight CO2 control. So let's look at a typical cell culture profile. In this example, pH is tightly controlled at 6.9, so you would imagine that CO2 is also staying relatively constant. What we see with continuous monitoring of CO2, however, is that pH doesn't actually prevent CO2 from fluctuating significantly. So even when your pH is controlled tightly, you can still have significant spikes in CO2, even if they're only for brief periods of time. These spikes can be compensated with base addition in bicarbonate systems, but even these brief spikes can be detrimental to the cell culture. The impact of CO2 on cell culture has been broadly studied and determined to have a harmful effect when higher or lower than the optimal level. When the CO2 is well above the desired concentration, of course, we know it's toxic for the cells. It's toxic because it slows the metabolic rate until the onset of apoptosis. But even if it's only slightly higher than the optimal level, this nonpolar molecule can still enter the cell and reduce the intracellular pH. This drop in pH affects metabolic processes, resulting in less productive cells, which is an effect that may go unnoticed if your CO2 is not controlled. And while insufficient CO2 is relatively rare due to the many sources of CO2 in the bioreactor, overstripping should still be avoided to prevent a deficiency in CO2's use as a reactant in other metabolic processes. Now, of course, this all makes sense in theory. By definition, you want the optimal amount of anything present in your bioreactor. But what is the tangible impact of a non-optimal amount of dissolved CO2? It's been well documented that even when CO2 spikes for brief periods of time, like in the previous example, it leads to lower productivity and decreased product quality. That can mean non-optimal glycosylation, it can mean a reduced therapeutic effect. But controlling dissolved CO2 can be used for more than just avoiding these negative consequences. It can actually be used to significantly improve your productivity. This example from a recent Roche patent shows a 100% increase in titer. That means double the amount of product between a process that's uncontrolled and an optimally controlled process. So there's a clear benefit in determining a golden profile for dissolved CO2 and tightly controlling to that concentration. Typically, this golden profile is established at the R&D scale. But in order to fully attain these benefits, CO2 has to be controlled to match that profile at all scales. This is partly because CO2 is much more efficiently stripped from smaller bioreactors than at larger volumes, which drastically changes the observed concentration of dissolved CO2 at the different scales. It can be difficult to achieve the high productivity reached by R&D at the production scale, and that's in large part due to variables like dissolved CO2. This disparity often requires many scaled down iterations to more closely match the desired performance. So by developing a control strategy for CO2 at each scale, this variable can be eliminated, and you can reduce the number of scale down iterations that are required to get the high productivity and quality achieved by R&D at the production scale. So it's clear that the control of dissolved CO2 is a critical factor in optimizing a bioprocess. Giovanni will now discuss the tools that are currently available for this measurement and what's coming in the market.
Thank you, Kelsey. I hope that you can see and hear me now. And thank you, Kelsey, again for refreshing us, reminding us why dissolved CO2 is so an important parameter. If we want to talk with the PAT language, process analytical technology language, we can call it a critical process parameter. So a parameter that you want to have under control. Now at Hamilton, we looked forward to have one critical process parameters more in our product portfolio to enable you to have really the control of your bioreactor. And we looked at dissolved CO2 after pH and CO2. And as Kelsey said, what is important is that you can control it. And the basis for a reliable control is that you can trust the measurement that you use in order to develop this automatic control loop. What do I mean by having a reliable measurement? Because of course, engineering speaking, reliable measurement in, good control out. What is a reliable measurement? Again, we speak about reliability. You need to trust your measurement. You need to rely on the technology that measures this parameter. It does need to have an affordable cost of ownership because it doesn't bring anything if you can have a very expensive technology to do your job when you cannot really afford it. And because we speak about control, and control for us means automated control loops, it has to be in real time because it has to go automatically. It has to be regulated automatically by the process controller of your bioreactor. Therefore, as Hamilton, we looked into the landscape and say, when we think about dissolved CO2, what do we find in the market before we introduced our own solution? When we speak about reliability, what comes in mind is most likely the offline method, so the blood gas analyzers, more or less. And probably you use some of them. There are different brands. And yes, they are the reference method, so they can be the benchmark, or let's say they can be used as the external reference, but you cannot really use them for real-time control. Even when you want, even when you do with the auto samplers, it will be simply too expensive. You know that they use cartridges, and even if you just measure every 10 minutes, the price of one single measuring point may be unsustainable. Let's just think how you can scale up to multiple bioreactors. So the reference method acts as the reference method. Maybe you measure that once per day. You think, okay, we need to have something that we can have that can have an affordable cost of ownership. CO2. Maybe in your bioreactor you see, but I do already measure CO2. Yes, you probably have an off-gas device that measures the off-gas from the headspace. And you can think, okay, then I can use these values and maybe I can do some mechanicistic model, or I can do some multivariate model, and then try to infer, try to calculate what can be the dissolved CO2. And in a very, let's say, clear situation that repeats itself, it may work. But if you, for example, start already to scale up from the smaller size bioreactor to pilot to production bioreactor, like Kelsey was showing us before, it's not going to work anymore. Especially if you will maybe compare what is the dissolved CO2 at the base of a 20,000 liter bioreactor is completely different of what you can have maybe in the head space. And you can find quite easily, if you just Google that in internet, research that shows that when you increase the rate speed of the agitation speed in your bioreactor, the off gas and then inline and dissolved CO2 will decrease. Therefore, you have the chance that if you use the off gas, you will tremendously underestimate the amount of dissolved CO2 that you really have in your bioreactor, creating or stressing your cells, creating all the detrimental effect that Kelsey was explaining in the previous slide. But there is also a real-time control, and someone of you may have seen it, may have used that. It's possible, or at least before Hamilton launched its product, so up to April this year, it was possible to measure dissolved CO2 in line with the Severing-Gauss technology. And probably you have it, and now we are going to discuss about that a little bit more in deeper, but we find out that it presents a lot of challenges. In short, we found out that the green spot, the solution that measures in real time, is as reliable or comparable in reliability, in accuracy to the offline method. And it has an affordable cost of ownership, was not really present into the market. And that's what motivated us at Hamilton to try to develop something on our own. So first of all, of course, we try to see, okay, there is already a technology. Can we make something to make it better? Can we improve the existing technology? And we looked into that. And what we find out, maybe you know that. So the electrochemical sensing of dissolved CO2 through the Severing-Gauss method. Severing-Gauss is the person who has invented the method more than 50 years ago. It's basically an adapted pH measurement. So what you have is a pH sensor put into a cap with a membrane, a gas permeable membrane, and the membrane is filled with an electrolyte, which of course is subject to falling. So what happens with this electrochemical technology for measuring dissolved CO2 is that the CO2 permeates to the membrane, enters into the electrolyte, titrates it, changes the pH of the electrolyte, and that's exactly what you are going to measure with this assembly. So it's a pH sensor with a cap, with liquids, and you know that pH sensor need quite some maintenance. In general, electrochemical sensor need quite some maintenance. Now, at Hamilton, we know that very well because actually we do produce and sell pH sensor, as well as we produce electrochemical dissolved oxygen sensor with a cap. And if you picture how to use this kind of assembly into your process, when your process, by process, we mean you have your sensor, you are autoclave or you sterilize it, then you use your sensor starting your bioprocess, and at the end you verify it. If you work with this kind of assembly, yes, it's a pH sensor, so you need to calibrate that before to use it. And then you need to make sure that it works so that the electrolyte is correctly refilled. From time to time, you need to change that this cap to measure it. So there are a lot of steps that you have to do in order to keep the sensor maintained. And it may be worth if in the end you get accurate measurement in line. The point is, and you can find it by the very manuals of these technologies, that when you put that in your process, you find out that after autoclavation or sterilization cycles, you need to product or the process calibrate this sensor because they drift after the first sterilization or after the first autoclavation. So in any case, you need to have the reference method to make sure that they are properly measuring. In short, there are too many steps. The steps that here are marked in red that we thought we really struggle with the idea that at Hamilton we can make it better if we keep using this same electrochemical technology. Therefore, we thought that to improve and to bring reliable cost-efficient real-time measurement, we got to change the paradigm. We got to change the technology. like what we did when we launched the first optical dissolved oxygen sensor. We wanted to come to this point. We wanted to cut down all the steps that are unnecessary from our point of view in order to deliver accurate measurement. And we were so proud, and we are still so proud after the launch in April that happened this year in April, because in the meantime, we have seen that we have quite some hundred of customers now using our sensor. So we see that what we thought, what all the effort we put into developing our sensor have come into place, and we see users working with it. We have brought in the market control, which means we brought a sensor that by its very name can enable you to have reliable measurement by which you can obtain reliable measurement and all the benefits that Kelsey explained, from R&D to production site, and eventually helps you improve your productivity and improve the quality of your bioprocesses. In the meantime, the same sensors and this sensor, I will say, what is peculiar of this new technology, even before I got into the detail and explained to you what is the technology behind it. What have we achieved with control? We have achieved a sensor which is maintenance-free. You just compare with what I just showed you about the severing gauss, and you see that there is no, it's a solid state technology. There is no liquid parts to change. You just have to calibrate it from time to time, and you calibrate that not as a pH sensor, but as a CO2 sensor, which means with air and gas standards, so with the parameter you want to measure. So it's maintenance-free, there is no replacement part, and it's calibrated easily with the parameter you want to measure. The measuring range that we have set up for this sensor is the one used for biopharma applications, so it's really a biopharma dissolved CO2 sensor. It has to be used in biopharma processes, so we did our best, and in the end, we even got the hygienic design certification. That means the sensor is cleanable according to the EHEDG class 1, so, and it is compatible with SIP and CIP cleaning. It's a solid state sensor. Again, compared to a sensor which has liquid electrolyte or liquid parts, it can be mounted at every angle in your bioreactor, which especially in the production of skids or in the production site, can be an advantage. And last but not least, the sensitivity of the sensor is so high that control measures only CO2. It may seem something like, okay, it's a CO2 sensor, so it's expected to measure only CO2. But for example, the severing gauss sensor I showed you before can titrate any molecule, any gas molecule that can go through the membrane, like ammonia also can also titrate the electrolyte. And therefore, you can have a measurement which is not only CO2. Now, control can detect exclusively and only the CO2 molecule. So you have a very, very precise and accurate measurement. And all these benefits enable us to be awarded. That was something that happens 1 month ago. So the pharma manufacturing platform and magazines awarded us the 2001 Pharma Innovation for the category monitoring and analytical devices. For the meaning that you see here, you can easily Google that and you can easily find that. If you want, you can have the link is there, and there is the QR code if you want to read the article. So the benefits that we brought with those sensor, that Hamilton brought with those sensor, beside the customer that are using that every day now since April, earned us also an award. Now, how do we came to have satisfied customer and even winning an award? Now it's maybe time to go into the technology a little bit more. So just not speaking about what you can achieve, but maybe how we have been able to achieve that. So what is control? Direct measurement of dissolved CO2 with a solid state sensor. What's the measuring principle behind control? Control is a solid state, is based on solid state middle infrared sensing technology. It's an optical solid state sensor. Now, if you are familiar with middle infrared theory. You know that middle infrared is based on the theory of energy exchange, of interaction. And interaction, we mean that there is an exchange of energy between two subjects. In the case of middle infrared, the two subjects are a radiation generated by a mid-infrared source, a lamp, and CO2 molecules that may absorb this radiation. If you're not familiar, don't worry, I'm going to explain to you right now. So in the middle infrared, you can irradiate a sample with middle infrared radiations, and you can collect these radiations through a detector that is specific for the molecules that you want to identify, recognize, or quantify. Now, what is very, very good for CO2 and middle infrared is that there is a single wavelength that interacts with the radiation, of which radiation interacts with CO2 molecules. That means that if you have a detector that can detect that specific wavelength, you are capable to see how much radiation or much radiation is absorbed by the CO2 molecules. In an easy way, if you just have a lamp sending middle infrared radiation and you have the detector measuring the CO2 and there is no CO2 molecules, as easy as that, 100% of the radiation goes from the source to the detector. Because there is no CO2 molecules, therefore there is no absorption. On the other side, if there is CO2 in the path length, the CO2 molecules, the second subject, absorb part of the energy, so interaction happens, exchange of energy, and less radiation reaches the CO2 MIR detector. And this can be used just to quantify it in a very direct way. And up to now, I have said that's no rocket science. That's known from decades. That's the basis of middle infrared theory, spectroscopy theory. So if this existed before, what is the innovation brought by Hamilton? Because that was already known. Well, the innovation brought by Hamilton was to put this concept into a 12 millimeter sterilizable or autoclavable sensor. Here is the measuring principle of control. That's exactly what we just explained. There is a membrane, FDA compliant. The CO2 molecules go through the membrane, got trapped into this membrane. There is a middle infrared source that sends a radiation, which gets reflected, goes through the membrane again, and goes to the detector. When the CO2 in the membrane is at the equilibrium with the CO2 in the sample, so when the partial pressure is the same, basically, then we can say that the difference in the energy detected by control is directly proportional to the amount of, to the partial pressure of CO2 that you have in the sample. And that was the Hamilton innovation. Put that all into a sensor. There is no liquid parts, and there is no part that gets degraded during the autoclavation and the sterilization cycles on cleaning cycles. It's as easy as that. Direct measurement of dissolved CO2 through meanings of middle infrared optical technology. So far, so good. I explained to you what's the technology behind that. I didn't explain to you up to now how accurate it is, because you remember, as we said, reliable measurement. How can we explain to you how reliable control is? Now, we wanted to be 100% sure that the measurement of control are reliable. So one easy test that you can do that we did during the development of our sensor was, and to be sure that there is no interference, we took one of the typical medium that you can have in cell culture, RPMI, and at certain specific time, we saturated this RPMI with using certified gas bottles containing different mixtures of CO2. CO2 at 5%, certified 5%, certified 10%, certified 20%, and so on. And we were sparging that and making sure that the the medium reaches the equilibrium before to measure. So in the end, what we obtained in this time was a target profile. Target profile of CO2, different concentration of CO2 into a medium that is used in cell culture. What does control measures when you measures in the same medium? That's how accurate control is. I think it's graphically evident what I meant, but if you want to know more in the detail and we want to speak about numbers, here is how we explain the accuracy of control. So you remember before I said that control works between 5 and 1000 millibar or 0.5 to 100% volume if we speak about volume. Now, between 5 and 100 millibar or 0.5 and 10% volume, the accuracy of control, absolute accuracy is plus minus 5 millibar. Above 100 millibar, the accuracy is proportional, is plus minus 5% of the target value. What do I mean by that? That means, for example, that if we consider 20% as the target value and 5% is the accuracy, relative accuracy at 20%, 5% of 20 is 1%. That means that the absolute accuracy of control at 20% is plus minus 1% volume, which means that the measurement of control can be between 19 and 20%. And it's probably the most accurate dissolved CO2 measurements that you can obtain in line at the moment. So that to explain to you what we wanted to achieve, when we said we wanted to deliver to you an accurate sensor. So that about the technology that we use, the measuring principle and the accuracy that you can reach with this solid state technology. But I keep saying it's a robust sensor. You remember feature that I said at the beginning, it's maintenance free. What does it mean, maintenance free in concrete? Now, again, in order to explain that to you, we needed to have a comparison. And as a comparison, we can take only the only other technology that is available to measure dissolved CO2, which is the Severing-Gauss one, so the adapted pH sensor. Now, if you assume 1 run per week, which is quite a lot, so to say, for 3 years, and if you scale it, you can imagine that we are speaking about 150 runs in 3 years, and 1 runs is the one that I showed you before. So autoclavation or sterilization, process, CIP, and then all the rest. That is the costs that you have to sustain when working with the Severing-Gauss technology and the cost that you have to sustain when working with the control optical technology. In short, with control, you only have the initial costs because you don't have to replace any part along the entire life cycle of the sensor. There is no spare parts. You just have to calibrate it from time to time. You don't have to replace a membrane. You don't have to refill the electrolyte. And you don't have to change the inner body pH sensor, which is something you need to change with the Severing-Gauss technology. So if you consider the cost along these 150 cycles or 3 years, if you want to call it in another way, you see the difference. So it's not just probably the most accurate inline dissolved CO2 measurement technology you can have, it's also the most cost efficient. So much about its robustness. Last but not least, if you know Hamilton, you know that we have been the pioneer of the microtransmitter technology, or like we call it, ARC technology. Control is a digital sensors. That means that it embeds the ARC technology. That means that it has the embedded ARC microtransmitter. You don't need an external transmitter to operate control. Your digital signal can go directly from control to the operating to the process operating system of your PLC of your bioreactor with a digital signal, which means that you don't have any problem like any other analog signal. And in control, you have all the functionality of a transmitter. That means you can have all the calibration data, You can have all the warning and errors indication. We have a built-in quality indicator. So everything is built within the sensor. If you follow Hamilton, you know that the main advantage of this concept is that you can calibrate the sensor in your laboratory in quiet, and you have your measuring loop, and then you can take it in production and just use it without needing to calibrate that directly in production, because the measuring loop is here. The sensor plus the transmitter is built in the same technology. The standard communication that we have, it's an open system, so it's a Modbus communication, so RS485, and you see it's a VP8 connector. So that's what we have as a standard. But of course, we want to have a sensor that can be easily implemented in your processes and can be programmed. So beside the digital, the Modbus that I just explained, you can have the compatibility from now on with OPC UA. So we do have an OPC UA module that you can plug to control so that you can transfer the measurement of control directly to OPC UA. We have an easy connection when you test the sensor with the PC, so that you can configure, calibrate, and trending. So you just have a USB cable that you plug to the sensor, and you can use to do all the operation in the laboratory during the test phase or during the maintenance phase. And yes, maybe to have compatibility, retrospective compatibility with the existing sensor, where maybe the 4 to 20 milliampere is still the protocol that you use, there is also the possibility to have an adapter that converts the digital signal into a 4 to 20 milliampere signal, which we obtain exactly with this adapter, which is a ARC V2G adapter. This adapter consents you to convert the Modbus into a 4 to 20 milliampere signal, and it has another feature, which is this LED light. The LED light is connected with the quality indicator built-in control. Again, you see this sentence, ARC. Again, ARC is our proprietary technology by which we put the transmitter functionality within the sensor, which means that if you plug this adapter on top, you have the sensor that not only tells you how your process is going, measuring the dissolved CO2, it keeps telling you at every second how good it is measuring as well. I come back to the concept of you need to have reliable measurement. Now, the quality indicator tells you at every instant how good, if you can trust the measurement your sensor is bringing. It's a sensor that monitors your process, and at the same time, it has a diagnostic function that monitors itself. Reliable measurement once again, thanks to our ARC technology. And this head, if you want, has also the functionality that you can have a Bluetooth communication with our ARC Air software for diagnostic or for calibration, which is a software that you can have on your Windows computer, on your tablet, either it is Android or either it is Apple or your smartphone. So we try to make it compatible to as many platforms as it is possible. And since I mentioned ARC Air, for those of you who know that, and for those of you who don't know that, ARC Air is our software sensor. So our software to maintain the ARC sensors. It is a software platform that we have in two formats, basic and advanced. The basic version is for laboratory use, and the advanced version is for the production use, or if you want to call it in another way, is for the GMP use. Now, if you recall what Kelsey showed you in the beginning, we say that it's important in order to have the proper control of CO2 and to benefit of all the advantages of controlling CO2 that you monitor that at the R&D scale, at pilot scale, and at production scale. You cannot have a production scale without having a sensor that is, especially in biopharma application, a sensor that is not ready to be used and compliant with the GMP, like the Part 11. Now, the bundle control plus ARC Air makes your control solution ready for compliance with all the GMP regulatory, like the CFR 21 Part 11 or the EUDRALEX Annex 11. with all the features that you need to have. We talk about audit trail, we talk about user management, we talk about automatic production of calibration protocols and calibration reports, electronic signatures, and everything that is built in. Again, we wanted to provide you a solution that is not just ready to measure CO2, but that can measure CO2 in biopharma application. And in biopharma, the sensor technology is as important as all the routine and all the regulatory ready to go. And yes, as you see before, we have lower operating costs. That's our operating platform. So if you know ArcCare, yes, control is another ARC sensors facilitating your life, as well as our pH, our dissolved oxygen, and our conductivity ARC sensor. Control is one more of them. And as we just says, it has a lot of screens. It enables you to provide proper diagnostics. So as you say here before, just to give you a quick glimpse, that's how you can watch out if the sensor is working properly. Of course, if you have the adapter, you see green, everything is going good. But in case, instead of green, you see the yellow or red, you can just use your smartphone or your computer or your tablet, and you just go in nearby of the sensor, and then you can see, okay, why the status indicator gives me an indication. What is the quality indicator of the measurement? So 100% everything is going good. Otherwise, I got a warning message. And you have all the values that you may want to use in order to keep monitoring the status of the diagnostic of your sensor. Again, bundle of ArcCare plus control makes your sensor ready for use in the market, and not only at R&D scale, but also in pilot, and moreover in the production scale. Reliable measurement with affordable operational costs, measuring in real time. And as I just said, control is joining our ARC sensor family. Just to summarize everything that we just said, you remember before I said that before we thought that there was not the proper technology, the green spot that can deliver everything. Reliable measurement with an affordable cost of ownership, capable to measure in real time. We think that we have achieved that with control, and we should, if we look at all the customers using our sensor in the last month since the launch, so since last April till now, beside the award that we get, but I'm speaking really about getting the feedback from the customers, from the users. We maintain what we promised, and we are, as Hamilton, quite proud of it. And we hope that you now are listening will be now interested into try that on your own as well. Say that, as Kelsey said, now we are open for the question time. I'm not sure if Kelsey has already answered some of your questions. If not, we are ready to do that now.
Great, thanks, Giovanni. I did answer a couple of the questions, but there are a few that I think will be better answered by you verbally. So one question we had was about the response time. I know you touched on it a little bit, but can you speak to that more directly?
Sure. I think that, yeah, let me go maybe back to that. So in regards to the response time, is nothing that we have shown here quite directly, but it's of course something that we can speak about. The response time that we have is comparable with any other solutions that you have in the market. If we want to describe the response time with the typical criteria of the time to get the T19, so the time to go from 0 to 90% of your target value, can be 10%, can be 20%, we are about to 160 seconds, so to say. The sensor can measure in principle every seconds, and that's what the sensor does. So you get a measurement every seconds. But the time that you get to go from one level to 0, for example, to 20%, the T90 is 160 seconds. I hope this answers your question.
Yep, hopefully that's clear. The other question was about the lifetime of the sensor. Again, I know you talked about it a little bit, but if you wouldn't mind addressing it directly.
Sure. Kelsey is right. I talked about that indirectly. So when I say, let's assume in 3-year running cost, and I was telling you, well, let's assume 1 run per week. That means if you consider 50 working weeks per year, it's 150 processes. I was somehow also implicitly telling you what is the expected life of control. Of course, here we are speaking about what we have seen using the test protocols that we have during our sensor development, which are quite tougher compared to what you have in the market. I think that you expect that we test our sensor in tougher condition as the one that you can have. At the same time, you can expect that the world is so different and so various that you can have a lot of applications. I will say that we can expect in the range of compatibility of the application available for control more than 100 runs, up to the ones that you see here obtained with our own specifications, without changing spare parts. And the only thing you have to do is recalibrate the sensor from time to time.
Great. I think those are the only open questions that we have. We can wait maybe one more minute to see if anything else pops up. But overall, I hope that was very clear for everyone. Great job, Giovanni.
Thank you, Kelsey. So I said, maybe just one last word while you are thinking if you want to pose any other questions. If you have, beside this webinar, now you can pose questions, but of course you can reach out to us afterwards. And if you go into hamiltoncompany.com, process analytics, you can have in contact, and then you have find local contact. So we suggest you contact our local responsibles, and you can ask them. So that's in case you want, you are now, we hope that we interested you enough so that you want to contact our people on site, and feel free to contact them so you can organize a demo, you can ask more questions. So feel free to do that. That will be the best way to do that. Just in case some question, instead of popping out now, will pop out later.
Yep, absolutely. So no more questions have popped up. As Giovanni said, if you have questions, there's a lot of information on our website, or you can contact a Hamilton representative, and any of them are happy to help. So thank you all very much for joining, and hopefully you see the value in controlling your process with dissolved CO2. Thank you, everyone.
Thank you, everyone, also from my side. And thank you, Kelsey.
Yep, thanks, Giovanni. Bye, everyone.